that to understand, again, as an evolutionary biologist, if you have a really complex system, the way to solve it is you have to have a historical approach. You can't understand any of Jim's systems without understanding the history of that. Just like, and you can't understand a brain, which was not designed by an engineer. It evolved, jerry-rigged, moving through optimal minima all the way till it got to where it is today. And it turns out where it came from is hugely influenced by olfaction. And in fact, what do we know about the first brain? I think this is a really interesting question that this is what I'm trying to tackle, is can we use what we know about brains that live now to go back in time, triangulate to the first brain, and ask why did that first brain evolve? And I would submit that that would give us information about how the brain is organized and how it processes information, and it will give us organizing principles for our own brains, which, in fact, for all brains. In fact, that's what I'm going to argue, that all these brains, actually, even the honeybee is its brain, and some people are arguing, is actually homologous to our brain. It's the same brain. We've inherited it from common ancestor. Okay, so that's an outrageous claim. But what do we know about the brain? And well, the first thing we know is it evolved underwater, because life evolved underwater. And in fact, it's a very all the phyla that are present first evolved underwater. In this underwater world is defined by chemicals, and the first animals were single-celled animals that move in response to chemicals, that using chemotaxis. The first sensory system of any active animal, either a prokaryote, a single-celled bacteria, or a multi-celled animal, the first animals were only living in a chemical world where they're responding to chemicals. But it is now the use of chemicals, olfaction, for cognition is universal. It's used in all animals. So it's not only the first, it's also the most universal. It's the only universal sense. And across the animal kingdom, where it's been measured, and that includes Drosophila and nematodes and mammals, the largest gene family in the genetic code is devoted to olfaction. It's devoted to olfactory receptors, coding olfactory receptors. And it's not just true of primitive animals, it's of worms and flies, it's true of birds, it's true of humans. So that we're investing a huge amount into this system. So you've got this, it's the first system, it's ancient, it's universal, and it's still to this day under dynamic selective pressure, and it's being maintained at this very high cost to the system. It's also, in the world where it evolved, in the marine world, it is until the evolution of things like sonar, which was very late in evolutionary history. When we're talking about the first couple of 100 million years, olfaction is the only sense you can use for remote sensing. Even today, even though this is being sponsored by Minerva, vision is not as good as olfaction. And in fact, olfaction is an incredibly powerful sense, because you can detect a few molecules from a source, and you can detect that gradient. And what I'm going to argue today is you can do more than just simply move up and down plumes, but you can actually map where you are in space. And to get back again to Jim's talk, others have argued, computational neuroscientists have argued, that olfaction was the first problem the brain solved, and it was the hardest problem. And once you've solved how to map the world based on turbulent gradients and the unpredictability of the olfactory world, you can do anything. And then vision, anything else was a piece of cake. And it, and so what they're arguing is that really you do have to understand how the brain evolved in this world doing this. So, okay, so what do we know about the evolutionary history of the brain? Well, that's a problem because it all happened at once. There was an evolutionary Big Bang, in which this was the Cambrian explosion. And in the explosion, within 80 million years, suddenly we went from a world that just had little wormy things that lived in the algal mat near the ocean shore and did a little bit of burrowing into the surface, but basically they were all herbivores, and it was the peaceable kingdom basically. And then something happened, and within 70 to 80 million years, suddenly all the phyla, with one exception, all animal phyla appeared, and so by 580 million years ago, all the animal phyla, basically the ancestors were there, and then we've just all differentiated since then. What we're interested in is the understanding the evolution of life and understanding how cognition affects the evolution of life. So what happened in this Cambrian revolution is that suddenly there was no nervous system. Now you've got a nervous system. Well, what do you get with a brain? You get as opposed to a bacteria that just sees a stimulus and reacts. Now you can move beyond reaction. You can actually learn. Now, well, what's the advantage of learning? Associative learning is found in animals even without brains, animals with nerve nets. But the ability to learn means that you can predict, and if you can predict, you can control. And I mean, these are obvious things, but what I'm proposing is that other people have suggested the Cambrian explosion was caused by the evolution of learning. Well, I think we have to be more specific. I think it was caused by the evolution of the ability to map and navigate, specifically in response to odors. And I've got a more detail that I'm not putting into this talk, but there's a reason why, which maybe we can get at in the questions. But let me just give you the overview. You might not know that animals map, and this ability to map was first actually discovered at Berkeley by Edward Tolman, who was a great man, and who studied rats in mazes, and in 1949 published the paper, The Cognitive Maps in Rats and Men. And he fought bitterly with the behaviorists like B.F. Skinner his whole career, who believed that animals were stimulus response machines. But he thought, he had this outpost in Berkeley saying animals have representations, and Tolman has been vindicated, and his paper is actually continually cited even today, because it's realized that non-humans actually have very good mapping systems and could make they actually human and non-human cognition is not very different, is not so different after all. At the same time as Tolman was working at Berkeley, Gustav Kramer in Germany was studying the carrier pigeon, the homing pigeon, and showing that homing pigeons also, their behavior when they're released from a place they've never been, the only way you could explain that behavior is somehow they had a map of their environment. It could not be explained by any kind of stimulus response thing. So by the end of the 40s, we had these two men who never communicated, both suggesting that animals, are making maps. And so we've got a mammal and we've got a bird, and I would say that since both of them, published this work in the 40s, both died in the 50s, both have been vindicated that there's now decades of research showing that both rats and pigeons are making maps, and making them in similar ways, which is rats in the lab and pigeons in the field. So really we've got, even though we've got the brains, birds and mammals separated 300 million years ago, we've got very similar cognitive abilities here. And recently, that circle has been closed by a beautiful study in the Egyptian fruit bat in Israel, where finally we're taking flying mammals and testing the same theories in flying mammals by taking these bats out to the desert where they've never been, just like a homing pigeon, and releasing it now, of course, with the advantage of GPS loggers, and showing that fruit bats navigate exactly the way you expect birds. You put them into a crater so that there are no visual cues, and they circle around and they take sightings, and then they decide, and then they take off, and then they beeline back to their roost, and they've never been in this area before. This is a baby fruit bat, and I put that up there because a new interest of mine is which is actually quite interesting, is the effect of cute animals on cognitive processing, which is becoming a really interesting field, the science of cute. I'm teaching a freshman seminar on this, and it turns out that you looking at that, if you think that's cute, which I think that is incredibly cute, but it actually, it changes, it activates your nucleus accumbens, it changes your focus, your attention, it makes you happier, it makes you slower and more careful if you're doing something on a completely abstract task. And you can get the same effect if I show you a car that's been morphed to have big eyes and a round face, just like a baby. So it's really interesting behavior. Okay, sometimes I throw these up in my slides in my talk. When my lectures for my students get really boring, I throw in a lot of cute animals. So. Okay, but pigeons. What was remarkable is that Floriano Papi showed in the 1970, and again, I keep using this word vindicated, but he fought for years against the other pigeon camps, and it's now the best studied system, the best studied sensory system in pigeons. And it turns out that pigeons absolutely need their noses to navigate. And if you block, so here's the loft, and you release pigeons from these 4 areas, and you just release them, and they all beeline back to the loft. And these are pigeons who have had their nostrils blocked, or they've various olfactory manipulations. And so this is really very strange. This bird has to have experience in the first 3 months of life living in a loft where its cage is open to the air, and so it can associate odors and wind. And if you block that, or if you can raise them in lofts with wind deflectors, and then they learn the wrong directions, and that will later affect their lifelong navigation abilities. So they're really sensitive, but once they learn their olfactory map, that's what they use the rest of their life. And this is an animal that is incredibly visual, but it's using olfaction, and in fact, there's no other sense that can substitute for olfaction, because if you knock out the nostril, they're lost. It's a very very interesting thing to see in such a long, such a visual animal. And in fact, carrier homing pigeons are bred for racing, and the racing breeds, the racing strains, actually have a larger olfactory system than the non-racing strains. So, but they also have a particular brain structure which they have in common with mammals, which is the hippocampus. And so here's a rat brain. This is a non-distorted drawing of the hippocampus and the olfactory bulb. So you kind of notice those are 2 large areas. The hippocampus is actually made of 2 pieces, an old piece and a new piece. The old piece is something that's homologous with lizards. The new piece is something that's only found in mammals and birds. And here's your hippocampus. Again, we've got all this, garbagey filing cabinets on top. But basically, to me, the important thing, this is the important part of the brain. This is where it all started. And in fact, the size of the hippocampus can be predicted by how much an animal uses space, maps space, and that's true of rats, pigeons, monkeys, humans, bats, fish, lizards, basically everything but amphibians, because no one's actually done the work in amphibians. And the size of the hippocampus, so the size of the hippocampus, and there's only been this one study in the homing pigeons, but the size of the hippocampus and the size of the olfactory bulb actually both are increased in birds that are specialized navigators. And so what I've suggested is that we have to rethink the olfactory system. We have to start thinking about it as part of a navigational system. And since I published that idea, primatologist has now published a new analysis of primate olfactory bulb and hippocampus, and showing, just like in the homing pigeon, the olfactory bulb and hippocampus are predicted by home range size in solitary primates. So what that means for our brain is, I think we really have to think differently about the limbic system, which is considered this low-level emotional, kind of non-cognitive area. So there's the limbic system, the olfactory bulbs, the hippocampus, and the amygdala, and the hypothalamus. That's a rat, and that's human. And actually, it turns out that the amygdala, there are two parts of the amygdala. The olfactory amygdala actually also has low-level neurogenesis. So you've got these three areas, again, are the only areas where you have any kind of neurogenesis. And you've got similar areas, the antennal lobe and projecting to the mushroom body has the same relationships as the olfactory bulb and the hippocampus. And so what I propose is that this is the original function of this system was this olfactory navigational system, where it is encoding what, where, and why. And the what meaning the odors, but the idea is that you don't really know what something is unless you know where it is. And so these are intimately connected, and then the why is encoding the value of the stimulus. So that the first brain was making these crude maps where it was saying that here are some odorants, and this one is associated with something positive, this is associated with something negative, and so it's that's all the brain really needed to do, and the what, where, why, and then you can start making it more and more sophisticated and moving on to more and more interesting, with increased memory. And so if that's true, then there should be fingerprints of this system all the way through our cognition. And that's what, that this system should have, if it was the first operating system, the olfactory navigational system, then there should be ways that can help us understand human cognition. And that's exactly, I think you can make that argument. Marcel Proust's Madeleine, what was, is an olfactory memory. And what's interesting about Proustian memories, which are now being studied in great detail, there's a whole new field of olfactory cognition, is that they are more emotional. The olfactory memories, if I tell you, recall the memory of, let me not go into experimental details. The reason you remember more clearly in response to olfactory cue is because olfactory things that are encoded with an olfactory cue are encoded with more emotion, and there's just more emotional activation. And so that seems like that's fine, but that's very primitive. But what's interesting is that they're also they trigger clearer episodic memories, which an episodic memory is by definition only found in humans, and it's a spatial temporal memory. It's a memory of right now. You've never seen me give this talk before in this slide, so you might have a visual image, and if you right now were smelling something unique, and tomorrow someone gave you that smell, or 10 years from now, you would You would smell that, okay. And so I would argue that here the most human of all memories, which is episodic memory, which is autobiographical memory, has this deep olfactory foundation, and so and that's just the tip of the iceberg. And we really need to think about human cognition in terms of olfaction. And I also study squirrels, and we do study squirrels. And I'd like to thank my funders and my graduate students and postdocs for their help in helping me with these ideas. Thanks.